Good with matter and elements and atoms and the basic chemistry definition? Cool. All right. So what are our 4 major molecular models? These are all organic. What do we got? What are our 4? Carbs, carbohydrate. Good. Lipids. What else do we have? Protein. And nucleic acid. Nucleic acid. What makes up protein? Amino acids. What makes up nucleic acid? Nucleotides. There we go. How about lipids? The fatty acids. Really, those are the individual chains. Fatty acids. And carbohydrates. I'm not a huge fan of the structure for that. Obviously, we have polysaccharides, which are built up from monosaccharides. So monosaccharides are your simple sugars. So if we have polysaccharides, we're going to have a chain of them. Okay. Sweet. Any questions on this stuff? No? So what is an example of a nucleic acid? I promise you needed to survive if you didn't have it. DNA. So DNA is deoxyribose nucleic acid. So DNA is made up of nucleotides A, T, C, and G. Adenosine, so on and so forth. The specifics of that, we'll get into a little bit, but for the most part, that'll be genetic. What you guys doing? You should take it. You should take it. So similar to your carbohydrate chain, sometimes and DNA is double-stranded, so you end up with they're complementary to one another. A is going to be two. basically, you have a chain of those that builds up your nucleic acid, and each of these are your nucleotides. Make sense? Alright, we'll talk a little bit about functional groups. What are functional groups? What do you think? Any idea? What's that? What do you mean? Functional groups. The functional groups are the reactive parts of organic compounds, and they help determine, drive how a compound will react. And what reactions that particular compound undergoes. So one example is in this case, R is just the rest of the molecule. Nitrogen and hydrogen, we call this so back to our macromolecule discussion that we had. Do you know anything that might have this? Know any proteins or amino acids? So these are present in amino acids. And this is particularly important because most forms of nitrogen, outside of them being a part of amino acids, will kill you. So that's how we get our nitrogen, which is important to survive. Another really common is OH. This is our hydroxyl. It is a reactive group that's in a lot of things. We'll talk a little bit about reactions. This is important in hydrolysis and dehydration reactions. Where do you think we see OH groups? It's sort of a pivotal example of hydroxy group. Yeah. So that has to be acids and bases. But your most common example is an alcohol. So ethanol. That is ethanol. And the fact that it has that OH group is particularly important for the biological effects of ethanol. Okay. And the last one we'll talk about Biol4. No, this is phosphate. Um, this is a little confusing when it comes to chemistry because the oxygen is covalent bound to the phosphorus. But together it makes an ion. That's why we use phosphate. What is a good example of a phosphate group? How many of you talked about this in high school biology? And probably your seventh grade biology class. You definitely covered it in seventh grade. What's that? Phosphoric acid, yes. In your body, hopefully not. Keep your insides out. Adenosine, diphosphate. ATP is particularly useful for energy storage. So the phosphate bonds have a lot of energy. But it's also important for signaling mechanisms in the body too. We talked a little bit about nucleic acid. We can have A, B, C, and G. This is the same A. So this is actually a nucleotide with 3 phosphate groups. And similarly, you could have GTP or ATP, so on and so forth. GTP is used in the body a lot in cellular signaling. Any questions on this stuff? Wonderful. I would definitely not go above that. Once you take organic chemistry, then yes. Okay. Yeah, Biol131. Just the fact that you're in right now. Genetics will be far more applicable. Micro, we talk a lot about different types of bacteria. Small eukaryotic also not used for you for stressful. I would also satisfy your requirement to take virology if you really that fast. All right, lipids, carbs, and proteins. What do we use lipids for? Yeah, what do we use it for? Functionally in the body. Why do we want lipids? Why do we want fat? that's important, but think more macro or big picture, less cellular. What do we use lipids for? Energy storage. That's why we store fat. So, lipids, fats, oils, steroids, phospholipids, etc. Important for energy storage. Steroids are particularly important for cellular signal. Fats are contained both of glycerol and free fatty acids. Glycerol and free fatty acids. Your glycerol looks like this. And your fatty acids, the X structures, don't worry about it. Stuff like that. So these zigzag lines represent hydrocarbons. Each one of those first axis is carbon, and then it's full of hydrogen, but all the carbons have 4 bonds. This group here is called a carboxylic acid. Another one is functional. Carboxylic acid. And here are your hydroxy. So just like we had ethanol, but we had no way from the ethanol, glycerol has hydroxy group with glycerol. So these come together in what's called a dehydration reaction. That means we are removing water. So when these come together, we take OH and H, we end up with water, and those form a bond. We end up with our glycerol, with these new formed bonds. Don't worry about this. Now, we can have saturated fat as a unsaturated and saturated. Oh, what is it? Saturated fat are saturated with hydrogen. And so they lack double bond. Whereas unsaturated are not saturated with hydrogen, and so they have double bond. Saturated fats are solid at room temperature. Unsaturated fats are liquid at room temperature. So your vegetable oil is an unsaturated fat, but your butter is a saturated fat. Let's talk a little bit about why. If I have my glycerol and my 3 fatty acid chains, and I don't have any double bonds, they all fit together nicely. So they can pack closely together. If we pack it closely together, we'll have solid. And it will produce a more rigid compound. That's also important with cell membranes. So cell membranes are phospholipids. We'll talk about phospholipids in a minute. But if those phospholipids have fatty acids that are saturated, the membrane is more rigid. If it's unsaturated, the membrane is more fluid-like. That's important in organisms that live in cold temperatures. We have found that they have a higher proportion of unsaturated fatty acids to give that cell membrane fluidity. Unsaturated, we got double bonds, but that disrupts the cohesiveness of the compound. It doesn't fit together as nicely. So they're more loosely oriented, and so they are liquid. That's it. Okay. Phospholipids are very similar to our fatty acids. If this is our glycerol, we have our 2 fatty acid chains and phosphate. You may see this and especially in your membrane diagram. So this is our phosphate head, and these are our fatty acid tails. Phospholipids are important because they have a hydrophilic and a hydrophobic region. What is the difference between hydrophilicity and hydrophobicity? Salt water, salt water. Good. And if you didn't know or didn't remember, you can break down the word. So hydrophilic, hydro meaning water, and attracted to or love. An example that immediately comes to mind would be pedophilia. So a pedophile, ped meaning child, phile meaning but hydrophobic, hydro meaning water, phobic meaning fear or afraid or against. what's unique about these phospholipids, especially as they function in our membranes, is that we have a phospholipid bilayer. So our membrane has phospholipids on the outside and on the inside, forming a double layer. So that means that we can have water or aqueous solution on the outside, and we can have an aqueous solution on the inside. So we still have a barrier around the cell. You also will see steroids inside the membrane. Steroids, we said, are lipids. Lipids are typically hydrophobic because they're composed of carbon and hydrogen. They're non-polar. In fact, the chemistry of life dissolves life. So non-polar is not going to make water, which is polar. Any questions for this? Yes. Carbohydrates. What do we use carbohydrates for? As an example, carbohydrates we use for energy use and storage. We also use them as structural components. We have monosaccharides, disaccharides, and polysaccharides. Your monosaccharides are your simplest form of carbohydrates. They provide energy in the cell. Typically, they have 5 or 6 carbons. You have a pentose or a hexose sugar. Disaccharides have 2 of them. You have 2 of them, di meaning 2. Some examples of your monosaccharides. You have glucose, fructose. Those are going to be the most common ones. Your disaccharides, sucrose, and the lactose. Maybe 2 most common ones there. So if you're lactose intolerant, that's the disaccharide present in dairy. And polysaccharides are more for your macro energy storage. Those are your big change. Your most common polysaccharides are glycogen. The glycogen is chains of glucose, but also something like starch. And some examples of your structural are stiffness and cellulose. Chitin is more prominent in something like bacterial cell walls, whereas cellulose is in plant cell walls and produces the rigidity of the cell walls of plants. That's why I think your cell models, plants have a cell wall, rigid, where the animal cells just have the membrane. Cellulose is also an important nutrient. What do you think cellulose is? If it's common in plant cell walls, structural, and other things that we don't break it down. Think of what nutrient components build up in plants that have common barriers. Think of your plants that have prominent structural components, anything with membrane around it, so corn, beans, that sort of thing, all high in protein. They all have the structural sort of skin. And we have time. Lastly, we'll talk about proteins. So, proteins do a lot for us. They're very important. You wouldn't survive without them. Proteins help us perform transport. Signaling. Enzymes. So proteins, they say, have those amino acid groups. So you have your carbon, hydrogen, oxygen, and nitrogen. That is a big one. Nitrogen. And we'll talk more specifically about enzymes. So what are enzymes? What do enzymes do for us? Right. They are, we'll say, endogenous protein pathways. So that means endogenous means they are within our body. Proteins and catalysts. Catalysts help a reaction occur. So if we think of a reaction, we go from A to B. This is our what's called DA, or your activation energy. And writing it down, this is what's called a reaction coordinate diagram. This particular diagram, does it show an exergonic or an endergonic reaction? Exergonic or endergonic? What are your thoughts? We got energy on the y-axis, the process of the reaction on the x-axis. It's going to be an exergonic or an endergonic reaction. Well, that has to do with the catalyst. So right now, let's just look at the reaction itself. Exergonic or endergonic. So our energy level when we started is higher than the energy level that we ended at. So overall, it's released energy. If it's released energy, it is exergonic. The other way around, if we had something that looked like this, that would be an endergonic reaction. So what a catalyst does, it provides an alternate route with a lower activation energy. And that's why it helps accelerate or speed up the reaction. We can have catalysts that are inorganic, but in the body, at least with this section, we're going to be most interested in enzymes, which are protein catalysts. Does that make sense? Enzymes interact with a substrate via what's called an induced shift model. So when we first started discovering enzymes, we initially thought that they reacted as sort of a lock and key, which meant there was more of a rigid active site and a corresponding substrate. Their enzyme and its substrate. They interact here at what's called the active site. Our revised model of how enzymes interact with their substrates is called an induced fit model. What that means is it's less stringent on how it is done. Induced fit. If you put the wrong key in the door, it doesn't work. Induced fit, if it's a similar key and you try really hard and you shove it in there and you turn it a couple times. Does that make sense? Induced fit, it's less strict. We'll adapt around the substrate. Does that make sense? While we're talking about this, I'll talk a little bit about inhibition. So we can have competitive and non-competitive inhibition. So competitive inhibition occurs at the active site, whereas non-competitive occurs elsewhere. So in this case, competitive inhibition would have something bonding here in the active site, blocking the substrate in the enzyme. Whereas non-competitive, maybe we have a different reactive site over here. The non-competitive comes in here and interacts at that other site and causes a structural change to the enzyme so that it can no longer interact with the substrate. Does that make sense? Any questions on protein? The last thing that I want to talk about today are types of reactions. So over here, we talked a little bit about two different forms of reactions. We have our exergonic and our endergonic reactions. But the other characteristics of reactions that you want to know, we have hydrolysis reaction, dehydration reaction, and redox. We talked a little bit about dehydration reaction. So if we think of our fatty acids again, we have this carbon, which is bound to the OH, that interacts with this carboxylic acid group to remove a molecule of water and form that bond. So that is called a dehydration reaction, because we are removing water. Now, if we utilize the energy that we store in fat and go in and start breaking those bonds, the opposite of a dehydration reaction is a hydrolysis reaction. So in this case, these came together and we extracted H2O. We also could take H2O and add it into this and end up with adding our hydrogen and our hydroxy group and back up where we started over here. So it's just a reverse reaction. Redox reactions involve the transfer of electrons. You just chemistry that too. What? Did you come to my answer? No, I don't get there. And those are your major types of reactions. That's talking. Do you have any questions about anything that we covered? I know it was lost, even for seeing it a second time. What's that? Yeah, this is a little ahead of where you're at right now. Some stuff, but you'll get to it very soon.